cells, the building blocks of your body are each like a tiny universe packed with different levels of complexity. Cells have amazing properties of self-replication and regulation, using their DNA as a blueprint for making the molecular machines that carry out their different functions, called proteins. Countless scientific research papers have been devoted to understanding how cells work, and many different words have been invented to describe the strange phenomenon inside the cellular universe. In this series, we define 26 such terms from A to Z that are used in molecular and cellular biology. A. Acetylation. These terms that end in blankylation generally mean something is being added to a protein. Proteins are like molecular machines that perform different functions inside the cell. DNA, the genetic material, is ultimately used to make proteins. But after a protein is made, the cell can further fine-tune how a protein functions by adding something onto it. Think of it sort of like downloading an additional piece of software for your computer. Acetylation is the process of adding a molecule called an acetyl group onto a protein. Acetylation can change how proteins interact with each other and alter how they interact with DNA. For example, the cell stores DNA by wrapping it around proteins called histones. Histone acetylation can change how accessible the DNA is to being used to make proteins, and thus impact whether sections of the DNA called genes are active or not. B. Biotechnology. Like acetylation, biotinylation is the process of attaching a molecular group called biotin onto a protein. Biotin is attracted very strongly to another molecule called streptavidin. In the lab, this interaction can be used to help isolate, purify, and label proteins and nucleic acids of interest. In the cell, biotinylation also has many roles, including fatty acid synthesis and metabolism. C. Chip-seq. Chip-seq stands for chromatin immunoprecipitation followed by sequencing. So what does all that mean? Proteins can attach onto DNA and control whether or not it gets copied and used to make more proteins. An amazing vicious cycle. In order to determine what proteins attach to DNA and where, we can use chip-seq. So what is chromatin immunoprecipitation? Chromatin refers to the DNA in attached proteins. Specific proteins can be isolated using molecules called antibodies that are designed to attach to specific proteins, sort of like a fishing hook. This process is called immunoprecipitation. The DNA bound to the proteins you're interested in can then be isolated, and the DNA sequenced to determine the genetic code of the region where it's attached to the protein. D. Differentiation. We're often taught that DNA is what makes you unique, but what makes, for example, the cells in your eye so different from the cells in your skin? Don't all of the cells in your body generally contain the same DNA? How do cells go from one ball of similar cells to a whole bunch of totally different cells and organs? This process is called differentiation. Think of it like cells becoming different from one another. It's one of the biggest areas of study in molecular and cellular biology. Cells look and function differently, largely due to the proteins they are making. But how cells choose exactly when and where to start making all of these different proteins is a mystery. Cells like stem cells with the ability to turn into different types of cells are called undifferentiated and can be triggered to transform into different forms by many things, like the presence of chemicals or growth factors, physical cues from the surroundings and other cells, and likely other things yet to be discovered. E. Endocytosis. Cells can take in things from their surroundings by forming a pouch on their surface that surrounds molecules or other objects and sucks them in. One example of this is uptake of cholesterol. Endocytosis can be controlled by receptors on the cell's surface. Mutations that cause malfunction or absence of such receptors can, for example, cause the disease familial hypercholesterolemia, which causes high blood cholesterol levels and heart attacks early in life. But endocytosis has many varied roles, including transport of proteins. F. Frameshift Mutation. You've probably heard of gene mutations, but did you know there are a lot of different types of mutations? DNA is made up of sequences of molecules called bases. Groups of 3 of these bases are called a codon and form a 3-base code that corresponds to one amino acid. Proteins are made from sequences of amino acids. Anything that changes the codon sequences can potentially mess up the sequences of amino acids and the resulting proteins. One example is a point mutation, where one DNA base is changed to another. Another often more dramatic example is a frameshift mutation, which occurs when DNA bases or sections of DNA are somehow deleted or added in a way that causes an entire shift in the way the DNA is being read out as 3 base pair combinations, or in other words, the reading frame. This means that the entire rest of the sequence is potentially translated into different amino acids, and the whole protein changed. Think of it like changing the spacing between words in a sentence. Suddenly, the whole message can take on a different meaning, or even become totally not understandable. That was molecular and cellular biology terms A through F. If you enjoyed this video, please subscribe and stay tuned for the rest of this series on cell vocabulary A through Z. And remember,